In contrast to this group, which work was presented by Dr. Abramov, I work mainly in electrophysiology rather than molecular biology. So, in one of our projects, which results were recently published, publications is listed here, we worked we researched the signals induced by GABA. GABA is one of the most important compounds which act as neurotransmitters in our brain. GABA is an acronym from gamma-aminobutyric acid. So, how does it act? It binds to GABA receptors. After binding, each receptors open an ion channel permeable for chloride ions. Indeed, movement of ions through these channels can be registered as electrical current. So here you can see responses of single neural cell to the signal transmitted by GABA. So what did we find in that our project? We found that in granular cells in hippocampus, there are actually two subtypes of GABA receptors. One subtype is more or less classical, which acts after binding of GABA, and another subtype can open even without GABA binding. Receptors or ion channels of that subtype were shown to have quite distinct properties from classical receptors. Mainly, or most importantly, they have significantly smaller average opening time. So that was the point where we stopped in that project. Now, a couple of words in general. As far as I am aware, about 90% of research of Parkinson's disease in central nervous system is concentrated at a point where dopaminergic neurons are mainly localized. This is substantia nigra pars compacta. However, another part of brain, hippocampus, is first. It is a very important link in memory formation and cognition. And secondly, it was repeatedly reported to be severely affected at progressed stages of Parkinson's disease. So, after this project was finished, I continued some experimental work at a specific cell subtype in hippocampus, hippocampal granular cells. At that cells, I detected same receptors, same two subtypes of receptors, and I found that these GABA-independent receptors speed up the overall cell response to any signal. So this is exactly what can we see here. These lines involve the response of spontaneously opening GABA receptors, whereas these lines or these green lines do not involve it. Also, I found that the action of spontaneously opening GABA receptors depends on some unknown cytoplasmic factor. It's something inside the cell. When you remove a single receptor from the cell, it stops working. It is possible, however, classical GABA receptor still works when you remove it from the cell with a small patch of memory. So at this stage, I discussed these findings with Dr. Abramov, and we proceeded to the next point. So I tried to repeat these experiments on cells from PINK1 knockout animals, which is one of the classical models of Parkinson's disease. I found that these spontaneously opening GABA receptors have very small effect or no effect at all. And also I found that when I inhibited such an enzyme as protein kinase A or PKA, I got the same effect. Also, I looked into literature and I found that spontaneously opening GABA receptors can be indeed modulated by protein kinase A, that activation of protein kinase A reverses pathologies connected to Parkinson's disease, and that the loss of PK activity suggested to underlie several Parkinson's disease effects connected to memory loss and cognition impairment. So, having this in hand, what I would dare to say? Probably now we found previously unrecognized mechanism of impairment of memory and cognition in the course of Parkinson's disease. This mechanism is localized in a non-classical area of brain, not where 90% of people try to find such mechanisms. What does it give us in practical sense? First, if we look at this from the point of view of these receptors, GABA receptors are one of the most popular receptors in terms of neuropharmacology. There are huge libraries of drugs designed to act exactly on these receptors. Anxiolytic drugs, antipsychotic drugs, anti-epileptic drugs, and so on and so on. These two types of receptors display different effects in response to ligands, namely to different drugs acting on these receptors. So, if these receptors, spontaneously opening GABA receptors, are indeed one of the links in mechanisms, in a mechanism of memory and cognition impairment in Parkinson's disease, they become a very prospective target for the therapy with existing drug. Probably this is connected to the question which was already asked. Next, if we take a look from the point of view of this enzyme, protein kinase A, there are also a lot of drugs in use and a lot of perspective compounds modulating its activity. So they maybe can give us a clue to therapy of Parkinson's disease at advanced stages. And next, another observation which I made, which I had using PINK1 knockout tissue. So what do we see here? Here we see an electrical track of single receptor functioning. All these downgoing steps show the openings of a single receptor. So it is a specific technique allowing to cut out a single receptor from a cell with a patch of membrane and attach it to a micropipette. After that, if you approach with this micropipette a living neural tissue, and if this neural tissue releases some neurotransmitter, in this case GABA, in response to this neurotransmitter, receptor at your micropipette can open. So the frequency of opening reflects the concentration of neurotransmitter in this tissue which you approach. This is a kind of semi-quantitative way to measure the neurotransmitter concentration, because direct measurements is quite a hard task. So, okay, when I perform this experiment with the same membrane patch, coming to as close as I could to wild type tissue and to PINK1 knockout tissue, I detected different GABA concentration in them. Actually, I cannot say what does it mean. Maybe this observation can be used as an indicator for the Parkinson's disease development. Maybe, at least according to several observations, high concentrations of GABA in a tissue can be toxic for neurons. So maybe this is not only a hallmark, but also a mechanism of cell death in the tissue. But I have no much idea about, and I couldn't find anything in literature about possible mechanisms elevating GABA concentration in Parkinson's disease impaired tissue, and that's why I cannot go further in my hypothesis at this stage. So this is mainly it.
You tried.
So what we're getting at there, you can see, is coming back to the questions, it's not all dopamine. So one of the things that Sergey and Andre have done in the last 6 months or more is start to investigate other chemicals, other receptors, other neuronal molecules that are important and may be important for Parkinson's disease. Partly to understand the disease, but also, you know, if GABA receptors and their function are important in some of the other non-motor complications of Parkinson's disease, there are quite a lot of GABA modifying drugs on the market. Maybe some of those can be, as they call it now, repurposed for Parkinson's disease. You know, we don't know yet. I mean, that's quite a leap yet to go from a few observations to treatment. But I think, you know, we do need to be thinking, all of us as a field, are the molecules already out there? They've already been trialled in humans and have been put for pain or epilepsy or all sorts of things that maybe can be reused for Parkinson's disease. My last slide, which I didn't have time to talk about, was we've started this thought with cancer as well. There's many molecules around cancer that have not now used for cancer or never been used for cancer, but have been investigated, and they do things for cancer that cancerologists don't want, but maybe do things for Parkinson's and neurodegeneration that we do want, because cancer is an overgrowth of cells and Parkinson's is a loss of cells. And so you can imagine the molecular signals are the mirror image. And so I think I'm moderately optimistic that out there are molecules that we haven't yet tried that we could try in humans, that are safe because they've been trialled for some other conditions. So we know the toxicology, we know a lot of the pharmacokinetics and so forth. Yeah. So I'm reasonably optimistic about something. I've no idea which one it is. If I did, I'd have shares in that company and I'd be able to go on holiday for a long time. So should we stop for tea, coffee and biscuits? Thank you.